All right, you're welcome. So in this particular discussion, we're going to look at the production and flow of CSF. So in the previous discussion, we're looking at the ventricular system, which is shown here. So what we have now is a child that has developed hydrocephalus, right? So here we have a child with normal size ventricles, and on the other hand, we have a child with hypertrophied ventricles. So these ventricles are swollen because the flow of CSF is occluded somewhere. Same diagram, this one. Now, this is a net effect that we are seeing where the skull size increases. So remember, ventricles are embedded within the brain tissue. So if they're going to expand because the pressure is increasing there because the fluid is not flowing, that pressure is exerted on the skull. And since this child, the joints of the skull bones, which we call sutures, have not yet, closed, the skull will expand in size. And this is what we are seeing in these two babies. Obviously, in this case, it is severe because the occlusion, I'm sure, is significant because look at the size, of the child's head. That head, the size is even larger than that of an adult. Okay. So away from that, we now look at the cerebrospinal fluid itself because we looked at the ventricles. Now we have to look at the fluid that is found in the ventricles, the cerebral ventricles to be specific. So definition is a clear fluid. It has no color. It is colorless. Neither does it have any odor. Now, at this outset, we must know that CSF is a product of blood, more like glomerular filtration. Equally here, filtration occurs, but not as elaborate as in the kidneys. So it is produced by the choroid. These are special type of capillaries found within the ventricles, together with the cells that line the ventricles. Those cells are known as ependymal cells. So those cells are cuboidal in nature, or what we can call tau cuboidal, because at times they appear to be columnar. So you can call them as tau cuboidal cells. And if I can refer you back to epithelial tissue, you know the function of cuboidal cells. Cuboidal cells are highly associated with absorption, okay, whereas columnar cells are highly associated with secretion. So in adults, the total volume of CSF flowing in the ventricles is in the ranges of 100 to 150 mils at any given time. Then in a day, maximum 400 mils, that's half a liter of CSF is produced and reabsorbed back into circulation. So when you're talking about CSF, think of the ventricles itself, think of the subarachnoid space and the fluid that is there. So since it is a product of blood, at some point it has to go back to circulation. Similar with lymph. You know, lymph is fluid in the interstitial spaces. At some point, that fluid must find itself in the general circulation. Same thing with cerebrospinal fluid. So you can think of cerebrospinal fluid as lymph of the brain. All right. So the amount of CSF produced is equivalent to the amount of CSF that must be reabsorbed. If there's any disturbance in the rate and the absorption, then you find that there's accumulation of CSF, and that way intracranial pressure increases. The pressure within the cranium increases, and that can cause, serious problems to an individual. One of them is herniation of the brainstem within the foramen magnum. Now, let's look at production, or rather secretion of CSF. Now, These are the capillaries found in the choroid. So that is the lumen of the capillary. Just follow the pointer there. Then these are the squam cells forming the walls of the capillary. Look at those flat cells and their flat nuclei. Okay, then the space there is the lumen. Now, capillaries are single-layered vessels. So there is no medial, there's no external. It's just one layer of squamous cells. Now, these squamous cells are held together by zonular or tight junctions. Zonular occludens or tight junctions. So, connecting this cell to this one here, you're going to find very tight junction. And now, tight junctions do not allow seepage of any fluid or ions whatsoever, right? So the only way that material that is flowing through these blood vessels can leave is via transportation across the cell membrane of these flattened cells. As you can see, that arrow. So this fluid or ions cannot pass in that direction because they are tight junctions. They seal off. That's why they are called zonular occludens. They occlude any space that may be potentially possible. So the first process in the formation of CSF is actually active transportation of sodium ions. So that arrow you're seeing there indicates the direction of sodium ions. So at any given time, you know that extracellular sodium is greater than intracellular sodium in terms of concentration. Okay. So sodium is essential for brain cells because nerve impulse conduction is dependent on the normal concentration of sodium in the extracellular space of the brain tissue and the normal concentration of potassium inside those nerve tissues. So the interplay, the migration of these cells or the transportation of these cells, of these ions across the cell membrane of nerves is what we call action potentials. Right. So these tight junctions together with a layer of pia matter that you can see there, this whitish layer is pia matter, then tight junction. So this layer, this thickness of flattened cells and pia matter and these ependymal cells, look at those tall cells with their nuclei located at the base. These are the ependymal cells. So this one, flattened cell, pia matter, as well as ependymal cells, create what we call the blood cerebrospinal fluid barrier. So cerebrospinal fluid is accumulating here. This is blood. There are blood cells in there and all the elements found in blood, inclusive of waste, hormones, and anything else that blood is able to transport is flowing here. Now, this is where the ventricles are, the spaces. Then this is a choroid. So what will be in this space of the ventricle is cerebrospinal fluid. So this is a barrier. Blood, cerebrospinal fluid, barrier. So that's the first process or first step in the formation of cerebrospinal fluid is active transportation. And this requires energy to transport or to pump out sodium ions from the lumen of the blood vessels into the ventricle. Then what follows now is attraction of chloride ions as well as water molecules. So these others now follow by passive transportation. So water molecules together with chloride ions will now follow or migrate towards sodium ions. So they are now accumulating here in the ventricles and become part of cerebrospinal fluid. So two processes are involved there. Number one is secretion, then followed by, number one is active transportation, which is a transportation of sodium, then followed by passive transportation of water molecules and chloride ions. Okay. Now, this is the normal composition of cerebrospinal fluid, all right? So appearance, we're saying it's clear and colorless. The pH is neutral, that is 7.4. Total volume, 1 to 150. Daily production is about 500 mils. Specific gravity, protein amount, so on and so forth. So this is the normal composition of CSF. So we see that cells have no chance. White blood cells, red blood cells, platelets, thrombocytes, all those. So of them, it's the platelets that are very minute. They are the smallest. So even them cannot pass through those tight junctions. It's not possible, right? So there is no white blood cells in there, okay? So unless there is infection, maybe by viral or bacterial causes, or indeed fungal infections, then we can find some immune cells in the cerebrospinal fluid. That's why a lumbar puncture is an essential tool to assess the integrity, or rather the infection status of the brain, whether it is still sterile or there is some infection there. So maybe pus can be found together with microorganisms and immune cells. What is the function of cerebrospinal fluid? So it acts as a cushion. So this is a brain, this is a spinal cord. So the fluid is circulating here in the subarachnoid space. So the brain and the spinal cord is floating in cerebrospinal fluid. So that is a cushion. And at the same time protects the central nervous system, which is brain and spinal cord. Next is provides mechanical buoyancy and support to the brain. So buoyancy, we're talking about ability to float. So the brain is made lighter because it is floating in water known as cerebrospinal fluid. Okay, so that way it becomes easier to carry the brain wherever we are going, and it never gets tired because anything that is put in water is made lighter. Okay, it's much easier to drag a boat of the same weight than to drag a car that is stationed on ground. When the boat is on water, it's easier, you can pull it alone. But the car of the same weight on the ground and its friction, you find it becomes very difficult to pull that. So that way the brain and the spinal cord is made lighter. Okay. Next is that serves as a reservoir and assists in regulation of contents of the skull. Mainly we're talking about the microenvironment of the brain. Microenvironment of the brain. So we're talking about micromolecules of the brain. Now, those micromolecules are essential for the normal function of the brain. We're talking about ions and electrolytes that should be present in the brain tissue. So because of those tight junctions found in the capillaries of the choroid plexus, we find that there is regulated amount and type of molecules that can find itself near the brain cells. So as a result, there is regulation of micromolecules in cerebrospinal fluid. Then removes metabolic waste. Okay, when nerve cells are done with their metabolic functions, waste will flow in the cerebrospinal fluid, which later on connects with venous blood. Same with lymph. Lymph also is connected with venous blood. Then it has to be taken to the heart. Then the heart pumps that blood to the kidneys for excretion of waste metabolites. Then lastly, it serves as a pathway for pineal secretion. Now, when we're discussing the endocrine system, we're saying endocrine glands secrete their hormones into blood vessels, to be specific, capillaries. And then those capillaries are connected to venules. Venules are connected to veins. Then the veins to the superior and inferior vena cava all the way into the right atrium of the heart. Now, When it comes to the pineal gland has no capillaries in which it secretes its endocrine hormone, to be specific, melatonin. So it simply secretes this into the subarachnoid space. Let me go back to that diagram. Yeah, that is a pineal gland. So the pineal gland will just secrete its melatonin here. And it will be collected because this is subarachnoid space. Then goes on like that. Then finally into the arachnoid granulations. Then enter the sagittal sinus. Then sagittal sinus all the way connected to the internal jugular vein, which is later on connected to the superior vena cava. So that's how melatonin will now enter the general circulation and affect the human body in one way or the other. So that is important because melatonin is essential in regulating the circadian rhythm, that is sleep and wake cycle. That is a gland there, pineal gland. Okay. So that is a function of cerebrospinal fluid. Let's look at circulation. So we have looked at production. Now let's look at how this fluid flows from the ventricular system and then finally merging with the venous system. So already just by following the dark arrows and the white arrows, you can tell the flow of CSF. The dark arrows are indicating flow of CSF within the ventricular system. This is the lateral ventricle, that is the third ventricle, this is the fourth ventricle. Then the arrows change here, they turn into white. This is now flow of cerebrospinal fluid out of the ventricular system and into the subarachnoid space. So all this is subarachnoid space. Then finally, again, they are back to dark. They have entered now. CSF has entered into the general circulation. So step by step, it's as follows. Okay. Just, I beg your pardon. I have some diagram to help us understand that. Okay, it's here. So first of all ventricles, that is lateral ventricles all the way to the fourth ventricle. These are areas where cerebrospinal fluid is being formed. Lateral, third, fourth. Secretion is occurring there. Then flow, since the lateral ventricles are the largest and they have the largest choroid, the amount of CSF they produce is greater than the rest, right? So it will accumulate there more than this. Then when it accumulates in the lateral ventricles, it will flow into the third, then finally the fourth. So we'll start with the lateral ventricles. From there, via the foramen of Monroe, or what you can simply call the interventricular foramen, that space there, cerebrospinal fluid leaves the lateral ventricles and enters the third ventricle, which is here. Then mixes with cerebrospinal fluid formed by the third ventricle, and then it flows through the midbrain. That is the midbrain. Through the midbrain, using a small tunnel or channel known as cerebral aqueduct of Sylvius, or you can call it mesencephalic aqueduct of Sylvius, that tunnel there, and enters now the fourth ventricle. From the fourth ventricle, we discussed last time to say there are about 4 foramen in which cerebrospinal fluid will flow out of the fourth ventricle. So we'll start with the lateral ones, the lateral apertures. So that is a lateral aperture. This is a median aperture. So lateral aperture are 2. One on each side of the fourth ventricle, otherwise known as foramen of Luschka. Then the median aperture is single and at the posterior wall of the fourth ventricle, or otherwise known as the foramen of Magendie. So through these foramen, the 2 lateral apertures and 1 median aperture. Cerebrospinal fluid leaves the 4th ventricle and leaves the ventricular system to enter the subarachnoid space. Now, the spinal cord has a central canal. You can see that, which is continuous with the 4th ventricle. So, some amount of cerebrospinal fluid flows in the core of the spinal cord using the central canal of the spinal cord. So, this cerebrospinal fluid in here does not leave the ventricular system. It's still there to reach at the bottom of the cord and then circulate back like that. So, it's a continuous loop where it goes and then back. Then it fills, then it leaves the ventricular system to enter the subarachnoid space. So once it leaves through this foramen, it enters the subarachnoid space there. And then through the arachnoid granulations. So arachnoid granulations, these are out pockets or invaginations of the subarachnoid into the dura mater. So this is a dura sinus. Okay, that's a dura sinus. And that is a dura mater. Look at that and that. So the white layer there is a dura mater. White layer there is dura mater. When we're discussing meninges, we said the dura mater that surrounds the brain has 2 layers, whereas the dura mater that surrounds the spinal cord only has 1 layer. So those are the 2 layers of the dura mater of the brain. And the space between them is what we call the dura sinus. Now, the dura sinus are only found in specific areas of the brain. One of them is between the longitudinal fissure. Come in. That is all in class. Right? All right, so I was saying the dura sinus are found in specific areas of the brain. One of them is in the longitudinal fissure, the fissure that separates the left hemisphere from the right hemisphere of the brain. So that space, the 2 layers of the dura mater split and form a sinus. Now, a sinus is similar to a vein because it has collection of deoxygenated blood. That's why it's shown blue here. You can see that is a vein already. That is a vein. So these veins are pouring there. They're collecting blood from the brain, going into the dura sinus. This is a sagittal sinus, to be specific, superior sagittal sinus. All right. Then also have a straight sinus between the cerebrum and cerebellum, straight sinus. Then we have a sigmoid sinus. There are so many of them, but all of these sinuses meet and form the internal jugular vein. And that vein descends besides our neck to go and form the superior vena cava and enter the right side of the heart, the right atrium to be specific. So those are arachnoid granulations. They are simply invaginations of the arachnoid matter into the dura mater. Okay. So they allow cerebrospinal fluid to enter the general circulation like that. So once CSF passes through the arachnoid granulations, it has now entered the general circulation. So what is here is deoxygenated blood, and it has to be taken out of the skull to go to the heart, because there are so many toxins now in this blood, because the brain cells have used up the blood and it's full of carbon dioxide and waste matter. Same with CSF as it flows. It is mainly composed of waste by this time. Okay. So this is a general flow of cerebrospinal fluid, and you must know step by step and be able to explain this scenario. So back to the arachnoid granulations. So these are, invaginations of the dura mater, of the arachnoid mater, and the PS, the dura mater. So that is arachnoid mater, and these swellings we are seeing, those are the arachnoid granulations. So these are the main drainage of CSF, so that CSF can leave the arachnoid, the subarachnoid space. So this is a superior sagittal sinus, which is shown here. This one here, and also there. That is a straight sinus. That is a transverse sinus. This is a sigmoid sinus. Then you see that perforation there. It's a jugular foramen. So all these sinuses, the superior sagittal sinus, inferior sagittal sinus, straight sinus, then transverse sinus, sigmoid sinus, all of them will come and merge here and then leave the skull as the internal jugular vein. So these blue structures are connected. And what you should know that these structures are more like veins. You can see that, except that they are so dilated, they are so wide, and they don't have valves like veins. Veins are valves that allow one-way flow, but these do not have valves. So that's why we call them sinuses. We can't call them veins. But the jugular vein has valves, and it's not within the skull for that matter. So all these structures you are seeing there are arachnoid granulations. Then that is a sagittal sinus. This is still the skull bone. That is a bone. Even though it is a bone, it has its own blood vessels. You can see those. Then this is a scalp and blood vessels in the scalp. Okay. Now here, what is being demonstrated is communication. All right. So this is a scalp. This is a skull, the bone itself. This is inside the skull, the dura mater and the dura sinus there, forming the sagittal sinus. So blood vessels in the scalp, the skin that covers the skull, will perforate the skull, the very tiny ones, and also reach the sagittal sinus. So we call such kind of vessels as emissary veins, more like messengers, allowing communication. So there's two-way flow of blood through these veins. So blood in the sagittal sinus can flow like that and reach the scalp. Blood in the scalp can flow like that and reach the sagittal sinus. So emissary veins do not have valves, just dependent on pressure. Which blood has more pressure? Is that if there's more pressure here, blood will flow out. If there's more pressure there, blood will flow in. Now, this is important because infection in the scalp, the skin, fungal, viral, and otherwise, even bacterial, or for example, there's an abscess in the scalp, and then maybe by any means that abscess ruptures, you find that that pus, together with the microorganisms, can migrate from the scalp through the emissary veins, reach the sagittal sinus, and infect the brain. Okay, where is that structure? Yeah, here. They'll be there and then flowing through the sagittal sinus and now connect back. So if there's microorganism there, what will stop these microorganisms to enter the granulations and circulate in cerebrospinal fluid, where it is circulating in the subarachnoid space and eventually infect the brain, cause encephalitis. So before it causes encephalitis, it will cause meningitis. So these meninges will be inflamed. The dura will be inflamed, the arachnoid will be inflamed, later on the pia, and eventually the brain itself. So that's why it's essential that these infections of the scalp must be treated carefully. So this is a arachnoid granulation after significant magnification. This is arachnoid, subarachnoid space. This is arachnoid mater. That is a pia mater. So here they're showing it as if the size of the pia mater and arachnoid is the same, it's not quite true. The pia mater is very thin. So space between pia and arachnoid is a subarachnoid space. This is where cerebrospinal fluid and blood vessels are found. Then this is the arachnoid granulation. So arachnoid granulation perforates the dura mater. You see that? This is dura mater. That is dura mater. There's nothing here. So there's now communication. So it allows CSF to pass through the dura mater and enter the general circulation to come here now. Okay. Yeah, to reach that. Now, within the granulations, the arachnoid granulations, there are no valves, right? There are no valves. So the only assurance that cerebrospinal fluid will join the general circulation is that at any given time, the pressure of CSF is greater. The pressure of CSF is greater than the pressure of venous circulation. Yeah, I think I have that somewhere. Yeah, here. CSF pressure is greater than venous circulation. So that's why flow of cerebrospinal fluid is usually in one direction, from the arachnoid space into the sagittal sinuses, or simply the dura sinuses. But sometimes, there can be some changes, maybe bleeding and so on and so forth, where the pressure is reversed. Now, when the pressure is reversed, still, fluid does not leak back into CSF. So when CSF collects there, it's not possible to flow back, okay, because the granulations act only as one-way channels. There's no way it will go back to the cerebrospinal fluid. So that's about the arachnoid granulation. So there are so many of them, but the largest ones and majority are found in the superior sagittal sinus, this part. Okay. Yeah. So there's so many sinuses. Most of them, this is where the arachnoid granulations are found. That one, the superior sagittal sinus, which is shown here. Superior sagittal, because of position. If you cut there, you are cutting the brain sagittal. Okay. This is right hemisphere. This is left hemisphere. Most of them are found along that. You can see that. Now we are saying these arachnoid granulations usually are found even in the fetus and newborn. So by 18 months of age after birth, they are visible even to the naked eye. If you take an MRI, CT scan, MRI or CT scan, or indeed, if you're looking at the brain of a cadaver of someone who is aged in this range, you'll still be able to see them. Now, as we grow older, these become calcified. So calcium is deposited here, just like we saw that calcium can also be deposited in the choroid plexus with age. So if it's deposited in the choroid plexus and the choroid plexus becomes hardened, we call that as a glomus. But if the arachnoid granulations become hardened, we call that as the Pacchionian bodies. So you need to be aware of those terminologies. Okay. So they have no serious, implications, except that the flow of CSF will be slower, but it will not cause any hydrocephaly or things like that. Okay. So that's about the arachnoid granulations. Now, the other structures to be mentioned for ventricular system and concerning the cerebrospinal fluid are cisternae. Now, cisternae are simply dilatation of the subarachnoid space. So they are different from granulations. These are granulations. But these spaces you are seeing, this wide space, it is not perforating the dura. It's just a space where subarachnoid space is increased because of the structure, the contours, and the shape of the brain itself. There are certain areas where they are large spaces. So we call those as cystern. Okay, but where the dura, the arachnoid matter PS is the dura mater. Those are granulations now. So you find that in this cisternae, there will be collection or there will be pooling of cerebrospinal fluid. So they are given names based on the area of the brain where they are found. Okay. So the area of the brain where they are found, then they'll carry that name. Okay. So they are simply dilatation of arachnoid space. All right. So you can see chiasmatic cystern. This is the optic chiasma, where the optic nerve crosses each other. Left and right, they are sending branches from each other and crossing to go to the opposite side. So we call that as optic chiasma. We'll discuss more about optic chiasma when we discuss the eye in detail. Then we have the interpeduncular cystern. So here we have peduncles, cerebral peduncles. These are more like large columns of white matter. Let me take you back a bit to, I don't think I have that diagram to be, yeah, here. So this is the medulla oblongata. This is the pons, that is the midbrain. Now, where these structures in the medulla oblongata, we call them pyramids. Okay, the ones that are anterior and biggest, we call them pyramids. You can even see them there. Now, the ones that lie lateral and posterior, we call them olives. So those are olives at the back and lateral, but the ones in front are pyramids. You can see that structure continuing in the pons. These are nerves, okay? The axons. Now, bundles of axons form nerves. Bundles of nerves form pyramids, like that. Now, in this area of the brain, we are calling them peduncles, right? Yeah. Here, we are calling them peduncles, connecting the pons to the midbrain. So that's why here we are calling it interpeduncular cystern, between two peduncles. Okay, that's where there's also dilatation of the arachnoid space, and you find the pool of cerebrospinal fluid. Then we have pontine cystern, because it is the lying juxta or adjacent to the pons. Then we have cerebellar medullary cystern. This is the cerebellum. This is the medulla oblongata. So that space between cerebellum and medulla oblongata, we call it cerebellar cystern or cisterna magna. Cisterna magna, because look at this. This is a skull. This is the occipital bone of the skull, and this is the base of the skull. You see that? Base of the skull. Even this is a base, because the occipital lobe plus the sphenoid bone and other bones help to form the base of the skull. Now, the base of the skull has a giant opening there, where inferior to this opening is spinal cord, superior to that opening is the medulla oblongata. So this is a level, if you cut there, superior to it is medulla oblongata, inferior to it is spinal cord. So that opening there is called foramen magnum. So that's why the cerebellar medullary cisterna is also known as cisterna magna. Okay. Then there are other, cisternae, but the major ones are about 6. We can just compare them with what is here. All right. So this is where cerebrospinal fluid is passing through all the way to the granulations and exit the subarachnoid space to enter the general circulation. Okay. So this is where we end with the ventricular system. Okay. As usual, if there are any questions, I'm available. We can have the discussions. Just reach out to me.